Hello and welcome to Nikolai's genetics lessons, and today's problem is excluding the amino acid glycine. All the other amino acids found within human proteins are, and you have to choose one of these answers. So let me remind you the structure of the amino acids, which has the same backbone and different R groups. So here's the structure. So here's oxygen, carbon, and another oxygen here. So we call this carbon carboxylic carbon, and this is carboxylic group. And here's another carbon, which we call alpha carbon. And here's another group, which is hydrogen group. And the third group of the backbone is going to be amino group. So nitrogen here and 3 hydrogens. So hydrogen, hydrogen, and hydrogen. So all amino acid has this backbone in their structure, and the only difference is going to be R group, which is here. In glycine, this is going to be hydrogen. Glycine is the most simple amino acid, so this is going to be only hydrogen in R group. All the rest amino acids, 19 amino acids, would have a structure here which is going to be more complex and would have more atoms here. Let me read the question again. Excluding the amino acid glycine, all the other amino acids found within human proteins are neutral at physiological pH. By the way, here we have positive charge and here we have negative charge. Depending on the pH of our solution, sometimes we may have 2 hydrogens here. In this case, we are going to lose this positive charge, and sometimes we can get a hydrogen here, and in this case our molecule is going to lose this negative charge here. But normally, under the normal physiological conditions, our amino acids are going to be in the Zwitter-ion form, so would have positive and negative charge, and we call such molecules neutral. So most of the amino acids are going to be neutral under the pH 7.4. 2 would be with negative charge, we call them acidic amino acids, and 3 are going to have positive charge, which we call basic amino acids, and they would have this positive charge not because they would have different charge here or here in the carboxylic group or amino group, but R group specifies the charge of the whole molecule under normal physiological conditions. So at the pH 7.4, 2 amino acid would have negative charge in the R group and 3 would have positive charge. The rest would be in Zwitter-ion form, would have positive and negative charge, which would cancel each other, and we consider such molecules being neutral. So the answer A, neutral at physiological pH, this is not correct answer. 15 out of 20 amino acids are going to be in Zwitter-ion form and would be neutral. Answer B, excluding the amino acid glycine, all the rest would be essential. This is not true. We have only 9 amino acids that's going to be essential, and 11 out of 20 would be non-essential, and glycine would be non-essential. It is very easy to memorize. This is most simple molecule, amino acid, and it's not essential. Very easy to memorize. And let me now skip variant C and variant D. Excluding amino acid glycine, all the rest would be optically inactive. This is not true. All the rest except glycine would be optically active, and only glycine would be optically inactive. So exactly opposite to this statement. Why we consider that glycine is going to be optically inactive? Because of this R group, which is the same as this hydrogen group here. In all amino acids, this carbon would be chiral carbon, meaning it's going to be attached to 4 different groups. So one group, second group, third group, and fourth group. But in amino acid glycine, this alpha carbon is not going to be chiral. We call this carbon a chiral because this carbon only attached to 3 different groups, 1, 2, and 3. And this R group is the same as this hydrogen group. One more time, excluding the amino acid glycine, all the other amino acids are going to be optically active. Only this amino acid is going to be optically inactive. So this is wrong statement, and we have only, now you see, correct statement is variant C. Take a look at this picture. So all amino acids can be in 2 forms, L form and D form. And those, they represent mirror images of themselves. They are not superimposable. For example, just like left hand glove, we cannot put on our right hand. These 2 molecules are not the same. If we put this hydrogen on top, for example, here, so we'll turn these molecules, you will find that they are not superimposable on each other. So molecules, amino acid molecules exist in 2 forms, L or left form and D or right form. All amino acids that we can find in proteins would be of the L form. For example, L-alanine, L-tryptophan, L-valine, and so on. But because in glycine we have 2 hydrogen groups here, hydrogen in R group, glycine doesn't have L or D configuration. Those, sometimes you can find that we call all amino acid being L form. Also you can find L-glycine, but actually this is not correct way to say, because glycine doesn't have no left or right form, no L or D configuration. So let me read our question again. Excluding the amino acid glycine, all the other amino acids found within human proteins are of the L configuration, and glycine just doesn't have no L or D configuration. Though sometimes again you can find in literature or textbooks or on labels L-glycine, though this is not very correct. So glycine just doesn't have no L or D configuration. And by the way, if in living organism we usually only can find L form, why we interested in D form? We can get this configuration when we artificially synthesize amino acids, and we don't want to get this form with our food because this may affect our health. This form sometimes can be found in some bacteria which produce toxic substances which we call antibiotics. So they need antibiotics for competition with other bacteria. And this is all for today. Thank you for your attention and see you in the next video. Goodbye.